Welcome back to The Explainer. Today, we're diving straight into something that literally sounds like it was pulled right out of a sci-fi novel, but I promise you it is happening right now in a lab in New York City. On June 1st, 2026, a preprint out of Columbia University sent absolute shockwaves through both the scientific and bioethics communities. What they achieved was the precise single-letter genome editing of human embryos. So the massive, unavoidable question we've got to ask ourselves right up front is, are we actually ready for precision-engineered embryos? Whenever we talk about tweaking the literal DNA of a future human being, the stakes are, well, unimaginably high. This new research forces all of us to confront this question head-on, blending these absolute marvels of modern biology with some of the deepest moral dilemmas we can possibly face as a society. Here is our roadmap for today's explainer. 1. A genome editing milestone. 2. The shadow of CRISPR's past. 3. The base editing difference. 4. Technical hurdles and mosaicism. And 5. The ethical divide. All right, let's jump right into section 1, a genome editing milestone, specifically that 2026 Columbia University study. Back in June 2026, developmental cell biologist Dieter Egli and his team successfully used a technique called base editing to alter the DNA of early-stage human embryos. Now, editing embryos isn't entirely new, but this specific tool absolutely is. Base editing is so incredibly precise, it's being hailed as a massive leap toward fixing disease-causing mutations before a child is even born. It's truly a monumental conceptual shift in how we approach human biology. Moving on to section 2, the shadow of CRISPR past. And yeah, a history of outrage. You really can't talk about embryo editing today without flashing back to 2018. That year, Chinese scientist He Jiankou used a first-generation editing technique called CRISPR-Cas9 on human embryos, implanted them, and 2 women actually gave birth to gene-edited babies. The global scientific community was absolutely outraged. The technology was widely considered entirely too risky and way too reckless for human trials. Because of that, He Jiankou actually spent 3 years in prison for illegal medical practice. So skipping forward to June 2026 with this new Columbia University preprint, it's a completely different scientific approach, but it undeniably lives in the heavy shadow of that 2018 scandal. Which brings us to section 3, the base editing difference, scissors versus pencils. So what exactly makes the 2026 study so different? Think of it as scissors versus pencils. First-generation CRISPR-Cas9, the tech used back in 2018, acted like blunt molecular scissors. It cut entirely through both strands of the DNA. Previous studies showed that doing this in embryos can cause the loss of whole edited chromosomes, which is wildly risky and cause severe unwanted genetic changes. Second-generation base editing, however, acts like a highly precise molecular pencil. It doesn't cut the DNA strands at all. It simply erases and rewrites a single letter of the DNA. It is vastly more reliable and totally bypasses that catastrophic risk of chromosome loss. The Columbia researchers took this molecular pencil and ran an experiment. Out of the roughly 3 billion, yes, with a B letters that make up the human genome, their goal was to find and swap just one. They literally just erased an A and penciled in a G in the genetic code of these early-stage human embryos. The sheer scale and precision of that is just mind-bending when you really think about it. So, how does this actually translate to real-world medical applications? The researchers targeted 3 specific genes. First, PCSK9. Naturally, this gene regulates bad cholesterol. By changing that one single letter from an A to a G, they essentially switch the gene off, which could massively reduce a person's future risk of coronary heart disease. Then they targeted HBG1 and HBG2. These genes are involved in fetal hemoglobin production. By making the A to G swap here, they mimicked a naturally occurring mutation that produces a highly protective type of hemoglobin. The ultimate goal? Protecting against devastating blood disorders like sickle cell disease and thalassemia. It is incredibly clever biological engineering. Next up is section 4, technical hurdles and mosaicism, or why this is definitely not clinic-ready yet. The primary reason this isn't rolling into hospitals tomorrow comes down to one tricky phenomenon: mosaicism. Imagine an embryo dividing into multiple cells. In mosaicism, some of those cells contain the newly edited DNA, while other cells retain the original unedited DNA. You basically get a patchwork, a mosaic. And when you're trying to fundamentally cure a genetic disease, you need complete uniformity, for sure not a patchwork. During the experiment, the team actually hit 3 major roadblocks. First, as we just saw, the edits suffered from mosaicism. They just weren't uniform. Second, to actually get the DNA editor into the cells, the team uses a snippet of mRNA, but at high doses, this caused cellular toxicity. And third, pushing those doses too high caused the embryo cells to completely stop dividing altogether. Lead researcher Dieter Egli has been totally transparent here. The technology is absolutely not ready for the clinic. If the tech damages the embryo in any way, it goes nowhere near human trials.
And finally, Section 5, the ethical divide, praise and alarm.
Even with those technical hurdles, proving this level of precision is even possible has ignited a deeply polarized reaction across the globe. On one side, we have significant praise. Greg Neely, a genomics researcher at the University of Sydney, said, "This will go down in history in a positive way, less reckless, more careful, and ethical than previous attempts." This really represents the optimistic view that this is a measured, responsible, and highly necessary step toward eventually eradicating severe genetic diseases before a child is even born. But on the exact opposite side, we have intense alarm. Stanford University biomedical ethicist Hank Greeley counters with a pretty severe warning. You could set up an IVF lab and a genetic testing lab for probably a handful of millions of dollars and start doing this. And one result might be really sick kids. The fear here is very immediate and grave. Affluent individuals or unregulated rogue clinics might see the study, think it's a ready-made blueprint, and just start offering base editing services prematurely, leading to tragic, irreversible health outcomes. And honestly, the criticism goes even deeper. Fyodor Urnov at UC Berkeley argues that editing human embryos is essentially a solution in search of a problem. Why? Because couples who carry severe genetic conditions already have access to standard IVF and genetic screening to select embryos without those conditions. Urnov warns that this research will solely impact the rapidly growing movement of embryo editors for purposes of baby improvement. He's highlighting the very real fear of a slippery slope straight toward designer babies. So, we are left trying to strike an incredibly delicate balance here. On one side, the goal, utilizing this incredible molecular pencil to safely eradicate devastating genetic diseases before birth. On the other side, the fear that proving this is possible will ignite a commercial gold rush toward unregulated baby improvement, with people seeking superior intelligence traits or physical enhancements. It's that classic struggle between unlocking profound medical miracles and opening a massive ethical Pandora's box. Which leaves us with this final, really profound thought to close out today's explainer. Where exactly is the line between preventing a disease and engineering a human trait? As this technology inevitably improves, society is going to have to decide exactly where that line is drawn. It's a fascinating and, frankly, slightly terrifying frontier. Thank you so much for learning with me today, and I will see you in the next explainer.